we kind of covered the DNA structure, and here's the stuff about RNA, right? So, RNA, ribonucleic acid. Remember, DNA was deoxyribo. This is ribo. So, no deoxy anymore, which means there's oxygen present right here. Okay? Instead of just hydrogen in deoxyribose, there's hydroxyl group in the ribose sugar. Okay? That hydroxyl group makes RNA extremely unstable. So when you work with RNA, there's a joke. If you look at it the wrong way, it's going to start falling apart. It's almost true because you have to store it in -80. When you work with it, you put it on ice. So it's really, really unstable molecule. And you may wonder why creating such an unstable molecule? What's the point, right? Well, think about the function of RNA. RNA, major function of it is to convey the message, right? To convey the genetic information from DNA to ribosomes to make a protein. And that if mRNA, if RNA molecules, does that make sense what I said? DNA, RNA, protein. So RNA is the middleman here. It's the sort of the courier delivery service. Okay, it delivers the message. And if mRNA will stay stable for a while, then protein can be synthesized over and over and over again, which is not really a good idea, because you can't then, it's really hard to control the synthesis then. However, if you get the message across, okay, protein is synthesized and RNA disintegrates, you can always make new molecules if you need more protein. You see the difference? Instead of trying to regulate the old ones. So this is one thing about RNA. Did we talk about the DNA stability and the plague? Yeah, so that DNA is very stable. RNA is not. You cannot find possibly any samples of RNA in things like fossils, even if we talk about 2 years ago. Okay, well, it's not a fossil yet, but I don't know, like corpses from people who died 300-400 years ago. Yes. It's a great question. I don't think RNA will sustain unless it was frozen in Antarctic, deep in the ice, because the coldest place on Earth is near South Pole. It's -80 Celsius. There is a place back in home country, which is the coldest place in the northern hemisphere, and it's -72 Celsius. So in Fahrenheit, something like about -90 Fahrenheit. It's pretty cold. And yes, it is true when people spit, saliva freezes before it reaches the ground. It is true. That can happen. One of my high school coaches was from there. So it's really an interesting experience. Anyway, so RNA is unstable. It probably would not survive. Although I would say it's the best place to dig for an old RNA. But since we have DNA that is much more stable, there is not really a lot of rationale to do that. DNA will give us much more information. Okay. Another difference between DNA and RNA molecules is that RNA uses different type of nucleotide, of the nitrogenous base. Instead of thymine in the DNA, it's going to use uracil. Okay. So those are two differences, but the pairing is going to be the same. So if you use uracil instead of thymine, it's going to pair with A. Does that make sense? Now, other implications is that RNA is single-stranded. You see it here in this illustration. So it's single-stranded molecule. but it can assume different shapes in the cell. Okay, those shapes, like this, are called secondary structure of the RNA. And the most common shape is this. It's called hairpin. This is the hairpin. This is the loop element. So hairpin loops are all over the RNA molecule, and they can play important regulatory roles. Think about it for a second. If you have an RNA molecule with a hairpin loop on it, and you have some sort of an enzyme that reads it through, or if it's a ribosome that tries to translate the RNA, make a protein. If it runs into such hairpin loop, it is likely to stall or fall off the RNA. Does that make sense? Just the whole idea, it's like a stop sign, like a red light. However, if you have some sort of another enzyme that regulates whether this hairpin is formed or not by modifying nitrogenous bases or cutting out pieces of RNA, then you can regulate whether the synthesis of a protein proceeds or not. Essentially, in one case you have the sign, road is closed, but if you can change this, then red light, green light, red light, green light. Does that make sense? These hairpin loops play important regulatory function. Of course, RNA, since it forms this internal bonds, of course it shouldn't say intracellular, it should say internal. That's my mistake. Internal bonds, it can be denatured. You heat it up, you denature RNA. Although, honestly, you heat up RNA, it just disintegrates. It's so unstable. Really. Now, there are 4 types of RNA that I want you to know. Doesn't mean that we know other types, but for the purposes of our discussions, I think those 4 types would be sufficient. Okay? The first one is messenger, mRNA. So M stands for messenger. You can see it schematically illustrated right here. Messenger RNA is the product of transcription. When DNA is transcribed into mRNA. And we will talk about transcription further, maybe 5, 7, 10 minutes from now. So far, it's mRNA that contains the message, the copy of a specific gene that is being expressed. Ribosomal RNA, which you can't see on any of those pictures, but believe me, it's a part of the ribosome, the structure responsible for protein synthesis. Ribosomal RNAs have both structural role, they kind of build up the ribosome, and functional catalytic role. Some of those ribosomal RNAs facilitate chemical reactions that happen during protein synthesis. One of the ribosomal RNAs is peptidyl transferase. It facilitates the addition of one amino acid to another in the process of protein synthesis. Okay? TRNA. The clover leaf structure that is shown here, you can often find this reference to clover leaf structure, okay? So it has 3 hairpins and 1 stem, okay? Stem is the site where tRNA, transport RNA, carries specific amino acid that is about to be incorporated into the growing protein chain. Okay? And one of the hairpins is called anticodon loop. Okay? This is anticodon loop. This anticodon loop is something that matches, okay, matches the amino acid, so it occupies the exact position in the protein sequence. I'm not going into very much detail about this because we will go when we'll get to the translation. Okay, if you look at this, that's the actual secondary structure of tRNA, and you can see that it has nothing to do with the clover leaf. But so it happened. There's a name, cloverleaf structure. The fourth type that I want you to know about is the microRNA, mRNA. MicroRNAs are small, double-stranded RNA molecules. They're fairly short, about maybe 18 nucleotide length. Now, numbers are cool, but I want to make a commitment and don't ask you any numbers, in the exam. So don't worry about those numbers. 18 nucleotide in length. Now this quite unusual structure, remember I told you here that it's a single strand molecule, right? The RNA. Turns out it can form double-stranded things, and this double-stranded mRNA, sorry, not mRNA, but microRNA, regulates the stability of the mRNA transcript. So it binds to the mRNA, okay, and this complex between microRNA and mRNA is recognized by the protein that is very conveniently called dicer. And dicer is acronym. I don't remember what it stands for, but it's acronym. Well, guess what it does? Well, if it's dicer, it dices, yes, it chops RNA, mRNA, this one, in pieces. Okay, so by destroying a specific RNA, and this recognition here is based on the sequence homology, okay, the complementarity, sequence complementarity. By controlling the amount of mRNA, you can essentially control gene expression. Now, the cool thing, microRNAs were discovered in like early 2000s, so maybe late 90s of last century. Nobel Prize was given something like 4 years ago. Very quickly, it was recognized as the major breakthrough, right? Now, why it's so cool? You can directly regulate the expression of a gene by using microRNA. You can actually design microRNA that specifically targets the very specific mRNA in the cell. Does that make sense? You can make it focused on, I don't know, mRNA that contains certain message. It is a research tool. It's awesome. You don't have to manipulate the DNA of, I don't know, of a bacteria or a human cell. All you have to do is to make microRNA, and you can synthesize it. It's not very expensive. Well, probably about $1,000 for synthesis and everything. Okay, and they will deliver you the reagent. You add it to the cell, and it turns off the expression of a specific protein. When all microRNA is gone, protein comes back on. That makes sense? If you destroy messenger RNA that is a copy of a certain protein, and this protein is not made, does that make sense? Eventually, this guy that regulates this destruction, microRNA, will be all gone. Protein comes back on. So it's not a permanent deletion of a gene from a genome. It's a temporary suppression, yes. And actually, it is reflected in the names. When we delete the gene from the genome permanently, we call it a knockout. Like, I don't know if you've heard knockout mice. You can, for example, delete the gene from the mouse that controls appetite. And mice become, AP2 leptin, mice become like this huge. They don't have the gene that gives them a sense of satiety. It's called a knockout. Does that make sense? And they're going to stay like this forever. It's gone from the genome. Or you can give them microRNA that will temporarily turn off, destroy mRNA of that gene. So as long as you give them that mRNA, they're going to become fatter and fatter. You stop giving it, they're going to shrink back. Yes. That's called knockdown, yes.
You know, these steroids, hormones, and antibiotics, like for cattle, over, like to make them bigger, things like that.
Yeah, but that antibiotics work in different way, and I can...
Anything to make it as big as they can get them, like the animals.
Oh, you can technically do it, but you mean using this?
Yeah.
You probably can, but depends on what you want it to achieve. Because if you want it to make the pig as fat as it can be, and I mean specifically fat, then yeah, you probably can treat it with microRNA, but the pork will cost a fortune, because unfortunately these treatments are so far extremely expensive. They rolled out some drugs based on this microRNA for the clinical trials, but they seem to be really, really expensive. It's really expensive just to make it, yes.
What's the hormone that inhibits muscle growth?
The cortisol inhibits muscle growth, for example. It's catabolic. It causes the...
There's like it's a disease I know that like you don't produce that hormone, so then you just like your muscles just get bigger and bigger.
Oh, wait, you don't produce myostatin?
There's a supplement called, like, it supposedly blocks that.
Myostatin?
Called MyoX.
There's like a deficiency that people actually don't produce it, so like their muscles just get bigger and bigger. I mean, they could always do that too if they wanted to.
Well, yeah, they can. Again, this stuff, the only problem, it's really expensive. That's the only issue. So antibiotics work better, yes.
So would you deliver this needle or would you digest it?
That's a great question. The digestion would probably not work because it's really relatively unstable. To my knowledge, it's either intramuscular injection or intravenous, but they usually also have a problem with delivering the drug into the cell because it has to go into the cell. So they pack it in all kinds of stuff like liposomes. Okay, so they coat it with the fat so it can diffuse from blood to the cell. And then there's another problem with targeted delivery. So you got to deliver it, sometimes you have to deliver it not to the entire body, but to the certain cells.
What if you deliver it to like stem cells and then designate them to become that part of it?
So if you deliver it to the stem cells, first of all, stem cells are spread throughout the body.
Well, I mean, like before, not in the body, but like culture cells outside and then.
With the stem cells, you can genetically manipulate them. So have you heard about CRISPR-Cas9? Pretty much you can cut the stuff out. You can cut the gene out and throw it away. And cell will be probably viable after that. So current technologists can do that. You can take, I don't know, bone marrow cells from a patient with a cancer, cut out whatever's bad with them, irradiate patient, get rid of all faulty cells, put those modified cells back into him or her, and they will reconstitute the normal blood now. It's expensive, and the problem with CRISPR-Cas9 that, as usual, first we discovered it. By the way, it was discovered, you know what it is initially? It's the bacterial immunity against bacteriophages, the viruses of bacteria. So it was a completely fundamental thing. Now, when they realized how they can use it in eukaryotes, oh cool, we're going to make a drug out of it. We're all going to be cured forever from everything. And then it turns out, it doesn't work as good as we thought it would. It just sometimes makes mistakes. Now, if you think about it for a second, if the drug doesn't cure diarrhea, it's sad, but okay. If the drug cures something and causes diarrhea, it's sad, but also okay. If the drug by accident cuts out the wrong part of your genome, that's not really a good outcome. I mean, you can have certain controls. They work on the controls, how to increase specificity and stuff, but it's still, I would say, for modifications of embryos, we're way far there. It's too far for us. In China, they already started doing that, but they take embryos, they modify them, and then they kill them because you don't know what you're gonna. It's ethical problem. Okay. So microRNA allows you not to mess with the genome, but again, you messed with genome once and you're cured for the entire life. This stuff you have to take on a constant basis. It's like a boxer, that thing knocks down. So a boxer will stand up again. When you get rid of the gene, you knocked it out. Okay. Now we discussed the connection between genotype and phenotype, and genotype is the entire genome that you have. Now, how big they are, those genomes? The smallest ones are generally in viruses. Then you have bacteria. Then you have fungi and other unicellular eukaryotes like protozoa and unicellular algae, and then plants and animals. Humans have the genome the size of 3 billion nucleotides. Okay. Smallest viruses, smallest delta virus, the smallest virus of the humans actually, is 1.7 thousand. So it's a huge difference. Of course, there are exceptions. For instance, mycoplasma, we mentioned it already, the smallest independently living organism, has the genome size of 0.5 million nucleotides. It's still bacteria. It lives independently. On the other hand, the Pandora virus has the genome size of 2.5 million, and it cannot live independently and is obligate intercellular parasite, which is kind of neat. Okay, so you see that there are variations. There are plants that have genome size, I don't remember the names, don't ask me. There are plants that have gigantic genomes, much bigger than human ones. Okay, that makes sense? So, but that progression is pretty accurate. In a general sense, you can set up, virus, bacteria, eukaryotes, unicellular eukaryotes, multicellular eukaryotes. Genotype does define phenotype, but as we mentioned, it's not all the genes, only the ones that are expressed at the given time. And while this concept may seem a little abstract, here's the great illustration of it. If you will grow the microbe Serratia marcescens, the one that we used for the transmissibility study, if you will grow this microbe at different temperatures, at the lower temperature, about 28 degrees Celsius, it will produce the pigment, and at the higher temperature it will not. Remember, we also noticed that when colonies become isolated and big, they produce pigment. And when population of Serratia is really dense, they stop doing that. So there are other environmental cues, environmental changes that change the phenotype. And this flow chart shows exactly this. Change in the environmental condition will alter the phenotype. It's rather hard to see on large animals, but fairly easy to see in bacteria, as we saw in Serratia. Right? Now the DNA, the genome, that is composed of all the genes. And what are the genes? It's not that easy to define. And I don't want to attempt it just yet. But gene is the part of the DNA that encodes something. Okay? So all the genes make up the genotype, they make a genome. And genome is in the chromosomes, essentially. Now, chromosomes in bacteria usually have one, rarely two. They're circular, which does not mean that they form a perfect circle. They just don't have loose ends. You see? That makes sense? Eukaryotic chromosomes are linear, and during this stage of the cellular growth, they assume a form of chromatin, right? Remember chromatin, the word? I want to say they assume the shape of spaghetti. Like spaghetti in a bowl. They're all tangled up and stuff. Okay? When cell divides, then they form that classic shape of a chromosome that you see here. It is still linear. Right? Both eukaryotic and prokaryotic DNA in the chromosomes, both of them are bound to special proteins. In eukaryotes, those proteins are called histones. In bacteria, they are called histone-like proteins, or sometimes nucleoid-associated proteins. Okay, so this is the place where bacterial chromosome is. We call it nucleoid. Now, my question for you is, why the hell are we wrapping up the DNA around histones? I can try and illustrate what I mean by wrapping up. I don't have any good thread-like thing, and I don't want to mess with the wires. Okay, well, imagine that I have DNA and I'm wrapping it up around histone. Okay, so DNA is wrapped around those proteins. What is the significance of that? Some parts of DNA are wrapped around, other parts being like unwrapped and be free of histones, and then they can be wrapped around again and then go unwrapped. What's the importance of that? Huh? So DNA changes structure, right? DNA that is wrapped around histones is different from DNA that is kind of free, unbound. When structure changes, what else changes? The F word. Yes. Right thinking. When structure changes, function changes. You tell me, what are the main functions of DNA? Storage of information, indirectly encoding for protein, expressing the gene, right? Now, storage is in the sequence. Sequence is not going to get altered, but expression can. Because for expression, you need to read the DNA. Does that make sense? You really need to read. Now imagine, you have a book. The book is on the shelf. It's standing on the shelf with other books. Can you read it, like from here? Or maybe even coming closer, like this close. Can you read it? No, you can't. What do you have to do first? Well, good luck opening the book while it's on the shelf. You have to take it. That's important. You have to take it off the shelf and then open. Well, guess what? When DNA is wrapped around histones, it's like a book on a shelf. You can't even open it. You have to unwrap it to be able to unzip it to read it through. Does that make sense? Now, you have different regulators that make DNA. I threw it. No, I didn't. You have different regulatory elements, different enzymes, blah, blah, blah. Some of them can do this. Some of them can do this. They actually modify those histones. Okay? So, one modification and it's all wrapped, other modification, it's all unwrapped. Does that make sense? And that will regulate which genes are expressed at the given time. You see? You make it loose, genes are expressed. You make it tightly bound, genes are not expressed. Right? Does that make sense? Now my favorite part. Well, bacteria are pretty tightly packed. Pretty much all DNA that they have are genes. Although not all of them are expressed and some of them are probably dysfunctional. But more so in eukaryotes. There's a lot of DNA that is called junk. DNA that does not encode for any proteins. Any idea on the percentage? Sorry, I'm doing that because I need to see that my microphone works. Any idea of percentage of junk DNA? What is the percentage of junk DNA in the human genome, for example? 30. So you said 30. Other guesses? Well, give me a number. It's not 4 points. 1%. Closest. How about 90? 90% of human DNA do not encode for any genes whatsoever. It sounds like a huge burden, but if you think about nature, not in philosophical sense, no, just nature is extremely bureaucratic and inefficient. But that inefficiency comes from the principle, don't break something that works. So if you have a gene that was functional sometime, but then stopped being functional, nature, some hypothetical, if we admit that there's evolution, okay, will not immediately force that gene to be cut out and thrown away. And let's admit we do the same. I mean, I have a bunch of broken headphones in the drawer. I don't throw them away, not because I really cherish them, just because I'm lazy. It's a different story. Well, in this case, this gene, this fragment of DNA that is not functional anymore, if there is no disadvantage in having it, it's going to stay there. Now, those 90% of junk, supposedly junk DNA, is not all of it. It's not all of it junk. Probably there are some fragments that have regulatory function and so on and so forth. Humans have a lot, as you can see. The tightest, the cleanest genome that, as far as I know of, is the pufferfish genome. It has almost no junk DNA. Don't ask why. You know what pufferfish is, right? And the coolness of it. And that the hypothesis that pufferfish is the cause of zombies. I'm not kidding. It produces the toxin, the neurotoxin, tetrodotoxin, which paralyzes respiratory muscle, blah, blah, blah. People die if you don't cook it properly. One of the hypotheses about, the whole zombie thing coming from Caribbean that I read was that pufferfish, the tetrodotoxin, was used by the voodoo dudes. I don't know how you call them, voodoo dudes. They gave it to people. I don't know how people accepted it, but that was not the point. And people almost died. So they had thread-like pulse, almost no respiratory activity. So for everybody who's not very, very specially medically trained, which is the most of population of Caribbean islands, they seemed like dead. So they were buried, okay? And due to the really being almost dead, their brains, circulation went down, so brain function got really impaired. And then later, those voodoo dudes got them an antidote, so those fellas almost literally raised from the dead, but due to the impaired brain function, they couldn't do a lot of things, except maybe don't kill, beat the hell out of somebody, don't. So that's a valid idea. At least it has some scientific background behind it. Now, DNA is not only in the chromosomes. In the eukaryotes, it's in mitochondria and chloroplasts. We've already talked about it. And it's one of the piece of evidence that suggests, that kind of proves that those guys were endosymbionts, right? In prokaryotes, the extrachromosomal DNA is in plasmids. And these are the structures that have enormous interest for us. Plasmids are double-stranded circular DNA molecules that can carry genes and do carry genes. These genes may encode a number of things. For instance, they may code for the enzyme that makes a certain nutrient available. Okay? So microbe takes an advantage of growing on oligotrophic media. They may carry a virulence factor. I don't know, something like microbe that causes respiratory infection. That toxin, that virulence factor will cause cough, and microbe will spread faster. Or it may carry capsule genes to protect against the immune system of the host. Or it may carry antibiotic resistance genes, which kind of sucks, because those plasmids can be transferred from one microbe to another. So if you have a microbe with antibiotic resistance in the population, this resistance will spread throughout. Does that make sense? This is the example of two plasmids that encode toxins. Now, I told you before, and I don't want to pretend that I make those awesome pictures. A lot of them are taken from that online microbiology book that I told you about. So this is a little bit of a misinformation here. You don't have to know the names of those plasmids, God forbid. So this PXO1 actually encodes exotoxin of the anthrax. This is Bacillus anthracis, okay, causative agent of anthrax. I don't remember what toxin does, probably kills the cells. They always kill cells. This one, PXO2, does not encode toxin. It encodes capsule, which makes microbe more resistant to immune system, and it survives. And anthrax can survive in the respiratory tract. You will not survive that. Usually 48 hours from infection to death, very rapidly progressing disease. Or in the digestive system, ulcerative enterocolitis, your intestines pretty much turn into like sieve. Or on the skin, so-called eschar. If you compare the Bacillus anthracis to its brother or cousin, whatever you want, Bacillus cereus, you see that it does not possess any plasmids coating those horrible toxins and capsules. So the outcome of Bacillus cereus infection is food poisoning. I'm not saying that it's a good thing, but it's definitely better than to die of the respiratory anthrax in 48 hours. Now, there are two processes, independent processes, that we're going to talk about regarding DNA. One is DNA replication. And this is function of it, okay? So DNA gets copied and and goes from generation to generation. So information is preserved and inherited. Does that make sense? Let's put DNA here and show two arrows from it. So one is replication. Another process is expression. What is the ultimate product of the gene expression? Huh? What defines, which molecule is the ultimate product of gene expression? You're right, it's phenotype. Protein, yeah. So you have to understand that replication and expression are two totally different processes. They not related to each other. Does that make sense? So every time bacterial cell divides, the entire genome is copied. That's replication. But during the lifetime of bacterial cell, different genes are getting expressed. And it has nothing to do with replication. Okay? Those are two things that do not overlap or cross over. Got it? Here you are, the expression. We're going to talk about expression first, and replication will be at the end. We're probably not going to be able to talk about it today. This is the flowchart that is called Central Dogma of Molecular Biology. It turns out it's not that much of a dogma, but in most organisms, this is the main route according to which gene information flows. It all starts at DNA. DNA is transcribed into the RNA. RNA is translated into proteins. Let's admit there's a lot of confusion when to use transcription and when to use translation. Is that so? I saw a lot. I saw phrases DNA is translated, protein is transcribed, and all other. I think I saw everything. This is the very, in my opinion, well, not maybe very easy, but reasonable way to realize what's going on there. DNA and RNA are written in the same language of nucleotides. Do you agree with me? What is the language of protein? If DNA is language of amino acids, yes. So DNA and RNA are the same language, but slightly different structure. So essentially, when you listen to the podcast or listen to the lecture, you can type it in as well. Does that make sense? You have audio file, you have a transcript. When you type the lecture, now those of you who take notes, you are transcribing my lecture. Does that make sense? That's transcription. It's still the same language, it's still English, although I have certain doubts about myself, but yours definitely is. Okay? So I speak English, you write English. Now, if you want to give your lectures to Japanese speaking person who has no idea about English, you have to translate them. Put it in different language. So during translation, we convert a language of nucleotides into the language of amino acids. Does that make sense? I will definitely ask you about it. To answer questions on the exam, you must know that DNA is transcribed into RNA and not the opposite. That RNA is translated into protein and not the opposite. Got it? As we just discussed, expression is defined by the environment, and the environment actually causes changes in the cell. It may be biochemical or physical changes that alter pathways, biochemical pathways, or change some regulatory elements. One of the best examples and easiest examples is response to some bacteria to the presence of lactose. What is lactose? Milk sugar. Generally, microbes, if you would take a microbe and start growing it on just TSA medium, those agar plates, they contain sugar, glucose. Microbes will use glucose as the food and energy source. But if you will put some of those microbes onto the medium that contains lactose, say milk, they will sense the presence of lactose in the environment and will start making enzymes that will utilize lactose as the source of energy and nutrients. And the way they do it is extremely simple and elegant. Lactose molecule binds to so-called repressor that prevents transcription. When lactose molecule, we're gonna go back to lactose operon later. I give you an example. I'm not gonna ask. By the end of the unit, you will have to answer this, but I want to give you a brief example. When lactose binds to the repressor falls off, and genes that utilize lactose transcribe. Does that make sense? When lactose is gone, repressor sits back on the DNA, and those enzymes are not produced anymore. Like 3-4 genes, it's called an operon, like 3-4 genes that produce enzymes that convert lactose. When there's lactose, they're made. When there's no lactose, they aren't made. They aren't expressed. That makes sense? Now, one gene. You have a gene, fragment of DNA. How many enzymes this gene will produce? That's not really an obvious question. 1, 2, 52. We now know, but I think it was in 60s when Biddle and Tatum did the experiment that granted them Nobel Prize later. They had a hypothesis of one gene, one enzyme. So that every gene produced one enzyme. And the experiment was pretty elegant. So they had a fungi called Neurospora crassa. Don't worry about the name here. And the wild type fungus was able to grow on the minimal medium that contained barely any nutrients, because most of those nutrients were produced by the fungus itself. And then they subjected Neurospora to ultraviolet radiation, pretty much making mutants. And they managed to select 3 different mutants. All 3 could not grow in the minimal medium. One required amino acid ornithine, another amino acid citrulline, and the third one required amino acid arginine. Now it turns out that ornithine, citrulline, arginine are all elements of the arginine synthesis pathway. Does that make sense? So arginine is made from citrulline, arginine is made from citrulline, and citrulline is made from ornithine. They were able to identify which gene has a mutation. And it turned out the gene that encoded for the enzyme that made ornithine, this gene was defective in a mutant number 1. Does that make sense? Gene that encoded for enzyme necessary to make citrulline was mutant in the mutant number 2. And finally, gene that encoded enzyme needed to make arginine was faulty in the guy number 3. Okay? So they showed that enzymes that are involved in arginine synthesis, and they already knew that there were 3 enzymes, that they are not encoded by one gene. You see what I'm saying? That it's not a gene for the entire pathway. Does that make sense? That each step, each reaction, each enzyme in this pathway is encoded by a specific gene. That was an elegant experiment. Biddle was working at University of Nebraska-Lincoln. They erected the building in his honor. I worked in this building for a year. It looks like a giant crematory with a huge chimneys on top. Very nice building inside. Awesome building. Great place. Now, well, we overturned a lot of things. Turns out this is not as simple. One gene may produce more than one enzyme. And we'll chat about it. One term that I want to introduce to you is the term oxytrophic. Okay, I'm going to mention this. It's a terminology thing. Oxytrophic are microbes that, for survival, must have a specific nutrient. Does that make sense? So all those 3 mutants are oxytrophic mutants. Yes. Yeah, they selected them. So they wanted, of course, they had probably many mutations. But what they were actually so they selected this pathway as the study object, because probably it was just they maybe had biochemical tools or something. Okay, it was easy. Yes. A certain nutrient, like this guy that needs citrulline, is oxytrophic, and this is oxytrophic, and this is oxytrophic. Okay, they all are oxytrophic. So they must have a certain nutrient in the medium. Of course, look, if we grow TSA, which is the protein from soy, partially digested with the trypsin, a bunch of carbohydrate, will oxytrophic microbes grow in it? What do you think? It has like all kinds of amino acids and shit. Sure enough, yeah. Because it has almost everything that microbes possibly want. Like blood agar, like brain-heart infusion. Think about this. Brain turned into like a mush and then dissolved in water, plus heart stuff. You're going to have there everything. Okay? So, oxytrophic, non-oxytrophic, who cares? Does that make sense? Right? No? It doesn't? Doesn't make sense? Okay. Back to humans. My son doesn't, one of my sons, well, both of them, they don't eat beans. They hate beans. Okay? Huh? I don't understand them either. So they don't eat beans. So if you take them to a party where all that is served is beans, they're not going to enjoy it very much, right? Does that make sense? Like, I hate onion. So take me to the party where all that is served is onion, I'm not going to enjoy it very much. But, or let's say, if you were a kid, sometimes, parents take you to a fancy restaurant, and if you're really picky, you're not going to be able to order anything because you hate all of it. But take the kid to the buffet, well, at least they have pizza. Does that make sense? So oxytrophic microbes are those picky guys. They must have something to grow. But if you take them to the buffet, which is any complex medium like TSA or brain-heart infusion or blood agar, something's going to fit. Does that make sense? Okay. So, transcription. first step of gene expression. Basically, you have an enzyme that is called RNA polymerase that consists of 4 subunits. You don't have to know the subunits: 2 alpha, beta, beta prime, and sigma subunit. Okay? That RNA polymerase reads DNA molecule and makes RNA. So this green line here at the bottom is RNA. And that's another green line here. This brown line, or beige, whatever, is DNA. How many strands of DNA can you see there? How many strands of, individual strands of DNA? 2. Okay, so DNA is double-stranded. How many strands of RNA can you see here? Just 1. So this is fundamental principle of life. It's actually as fundamental as the fact that, if people don't breathe, they die. Transcription. For transcription, RNA polymerase must have double-strand DNA. It's fundamental principle. One strand of DNA cannot be transcribed. There must be 2 strands. Does that make sense? Now, interestingly enough, there must be 2 strands, but only one is actually transcribed. You see what I'm saying? So you have 2 strands. One is called a template strand, and another one is non-template. Only template strand is transcribed. Does that make sense? So if you somehow artificially make single-stranded DNA molecule, it can be done. It's technically absolutely possible. It's not that hard. It's just expensive and takes quite a bit of time. But if you make the single-stranded DNA molecule, RNA polymerase will not be able to transcribe it. You have to make a complementary copy, and then you can start making transcripts. Does that make sense? So step by step, what happens? It reads DNA from 3' to 5'. Okay. First, enzyme interacts with a region that is called a promoter region. What is the function of promoter? What does promoter do? Huh? Yeah, so it says, hey, start over here. Promotes transcription, right? It starts. So that's like runners, they have a start line and they have some space before it. Like people who run 100 meters, they don't run like from the wall. They have some space before starting line so you can walk and stretch and so on. Does that make sense? Okay, so RNA polymerase binds to the promoter first. And then it interacts with 2 specific sequences. The sequence at -35 position and sequence at -10 position. These numbers, -10, -35, refer to number of nucleotide relatively to the first nucleotide of an actual gene. Does that make sense? So if this is start, then -1, -2, -3, -4, -5, for example. Okay, going backwards. It's not a gene yet. Those are 2 regulatory sequences. You can see, well, you probably can't, but trust me. These sequences have a lot of TA pairs. What is so interesting about TAs or AT pairs? Well, it's too complicated. What is the feature of what distinguishes AT from GC?
AT has 2 hydrogen bonds.
So they are weaker. They're weaker. Okay, they have 2 hydrogen bonds between the bases, and they are easier to split apart. So those parts here are probably the parts where DNA gets unzipped. You see? DNA gets unzipped here. There's no pairing. That's beige stuff. That beige stuff is the RNA polymerase, and DNA strands are separated here. I want to say that it's like running a zipper, but it is not, because when you open the zipper, you know, thing behind it stays open. Not in this case. You know, the RNA polymerase runs through the DNA, and the place where RNA polymerase is now is unzipped. But before it and after it, DNA is double-stranded again. By the way, that part, this direction is called, just FYI, downstream, and this is upstream. It's often referred this way. So okay, we've got our RNA polymerase sitting right next to the starting site. When the complex is made, DNA is unzipped, RNA polymerase starts to move on, producing, I don't know if you can see that, really, really tiny green thingy coming out of the mRNA. The first initial transcript is being made. The first few attempts are unsuccessful. It's called abortive initiation. An RNA polymerase slides about 10 nucleotides, makes short transcript, and falls off. Okay? It's like riding a bicycle. You're going to fall off first several times. But then eventually the complex is good, stable, and RNA polymerase continuously making the mRNA. Okay? That makes sense? Okay? That mRNA is made, and this stage is called elongation. Okay? Now, initiation, elongation, and finally, when RNA polymerase encounters so-called terminal repeats, pretty much a signal to end transcription, it stalls and falls off the DNA. You've got the gene transcribed. Does that make sense? Think about the terminal repeats and that initiation site as something that tells you where to start reading the sentence and where to finish. You see what I'm trying to say? You know that your sentence starts with the capital letter and it finishes with a period. Does that make sense? That's pretty much the idea. Last thing before we go to the break. We're going to chat about splicing. Now, most of the processes that we discuss here are in prokaryotes. But we do talk a little about some stuff that is unique for eukaryotes. You may ask, why don't we talk about eukaryotic, what we just saw, this one, is the transcription in bacteria. You may ask, why don't we talk about transcription in eukaryotic microbes? Because I have very bad memories of it. When I was in college, the prokaryotic transcription was 1 lecture. Eukaryotic transcription was 3 weeks. It has 3 different RNA polymerases, a buttload of regulatory factors, and honestly, if you're not going to work with it, you don't need that much detail. That amount of details is enough for me. Okay. Now, splicing. It is unique for eukaryotes. There is no splicing in bacteria or archaea. The junk DNA that we talked about is not concentrated on left 5' or 3' end of the DNA. It is interspersed through all the genes. So when you look at the sequence of a specific gene, you're going to find that it has fragments that actually encode the protein, okay? They are called exons, and fragments that do not encode, which are introns. When transcription takes place, the precursor mRNA, illustrated here, has both exons and introns, meaningful and rubbish fragments. And obviously, before you translate this mRNA, you want to cut out the rubbish. You want to splice them out. Think about video editing as an example. You take a long video. For instance, I don't know, you are filming. If you, I'm not hunting, so I don't know how it works. But let's say you're sitting hunting, okay, and it takes hours for the deer to come out, and you have a camera that's running all the time. Then the whole process of shooting the deer takes probably seconds. Well, maybe more than that, okay? Then you take the entire 4 hours and you splice out boring fragments. And what you left is what you're going to show your friends. Does that make sense? So here you have introns that are going to be cut out. There's special structure that is called spliceosome that consists of RNA and protein. That structures, the spliceosomes will cut it out. But here comes the idea of more than one enzyme from a single gene. These 5 exons can be all combined together. Can be all combined together like this. That's actual mRNA that, when translated into protein, produces this product. Just example, right? If you miss one of the exons, exon number 3, the product is going to be similar but different. Or you miss another exon. Again, product is going to be similar but different. You see? Does that make sense? Like with the movies. Sometimes they release movie that is shown in theater is one thing, and then they release director's cut, which is 1 minute 43 seconds longer. That's actual number from Paul Verhoeven. I think it's Striptease, something. Showgirls. I read it somewhere. That director's cut was 1 minute 43 seconds longer. They included some scene, whatever. So that's like different versions of the same movie, okay? Deleted scenes, undeleted scenes, right? Which essentially means that one gene can produce 3, well, 4, 5, whatever, different functional proteins, okay? Again, prokaryotes do not have splicing, right? But it turns out viruses do. Viruses do have splicing. Splicing was discovered on viruses, on adenoviruses. Not initially, because they infect eukaryotes, so they use a lot of mechanisms that eukaryotes employ. That make sense? Right. Let's take a break.